This is the first of a three-part lecture, Lecture 8, Regulating Gene Expression in Bacteria and Archaea. Most of what I'm going to talk about, though, we have learned from the bacteria, and I'll only briefly mention the archaea. This is Part 1. In Part 1, I'm going to give you a basic overview of protein regulation, and we're going to discuss concepts important in understanding pre-transcriptional regulation. A cell's genetic and metabolic machinery are integrated and interdependent. Enzymes necessary for metabolism are encoded in the genome. Thus, you need to express those genes in order to obtain the enzymes and then execute metabolism. The level of activity of enzymes must be carefully regulated, or metabolism will not occur productively, the cell will waste energy, and it will lose the ability to respond appropriately to the environment, including the loss of the ability to appropriately regulate at the divide and replicate at the right time and to the right level. This will then compromise the overall survival of the organism under consideration. So accordingly, we must regulate protein activity, either at the level of the protein directly or at the level of gene expression. Approximately 40% of the genes in any given cell are regulated, meaning that they are not constitutively or constantly expressed. Many others are constitutively expressed, but the proteins that they produce are then regulated at the level of the protein. Let's consider the levels at which regulation of proteins or protein activity can occur. At the bottom of this figure, so shown down here in brown, we have a cartoon depicting a protein. There's the methionine, the very first amino acid incorporated into all proteins. If this was bacterium, of course, that would be formylmethionine. Proteins can be phosphorylated, dephosphorylated, induced with competitive inducers or induced with allosteric inducers. They can be inhibited by competitive inhibitors or by allosteric inhibitors, and otherwise modified in a variety of ways, all of which affect protein activity. So there are in fact many ways at which we can regulate the functionality of any given protein. Moving backwards in the genetic message, or upwards on this slide, let's consider messenger RNA. So the messenger RNA, which is shown here in orange, can be controlled in a variety of ways as well. Many of those are methods that affect the stability of messenger RNA. So if you have a way to make messenger RNA more stable, then it lasts longer, or in other words, it has a longer half-life in the cell. If it lasts longer, then that means that it will be translated more frequently, resulting in more of the particular protein involved. If, in contrast, the messenger RNA is less stable, if it degrades readily, then it will have a shorter half-life, it will be less frequently translated, and you will have lower protein levels. Messenger RNA can also be inhibited by binding with what is called antisense, or small RNA molecules that are able to bind to messenger RNA regions where they are complementary, or where there is complementary sequence, thereby inhibiting the ribosome's activity. There's also a mechanism that I'll mention at the end of the third part of this lecture called riboswitches, wherein a modification to the messenger RNA itself can inhibit or block translation. If a messenger RNA molecule can't be translated, then you don't get the final protein, so that serves as a way to regulate gene expression and therefore regulate protein activity. Continuing to move backwards, let's consider transcription. Okay, so in green here we have the DNA, and so this act right here is transcription itself, okay, executed by RNA polymerase. Many bacteria and archaea regulate protein levels by regulating gene transcription. It's very, very common in the prokaryotic world. This can be done in a variety of ways, but typically involves pre-transcriptional regulation, which uses negative and positive control mechanisms that rely on repressor or activator proteins, as well as small molecules known as effectors. You have studied the lac operon, and we will review it here, so you already know a little bit about this type of transcriptional control. Let's consider the level of transcriptional control first, specifically the level of pre-transcriptional control. Regulating transcription typically involves the action of DNA binding proteins to sequences upstream of a gene. These proteins can act either to block RNA polymerase, in which case we refer to them as repressor proteins, or to promote the activity of RNA polymerase, in which case they are called activator proteins. Most DNA binding proteins that play a role in transcription of this nature have a similar protein sequence and structure. They tend to act as dimers, which is what this cartoon is showing. A dimer would be two proteins that are attached together. Okay? And they have regions on each that bind to major grooves in DNA molecules. Those would be these regions down here. And then at the opposite end of the proteins, they have regions that allow them to bind to one another. They are attracted to inverted repeat sequences as shown in this part of the DNA in this cartoon. So here are inverted repeats here and here. And if you look at them, and you'll see what I mean. An inverted repeat is essentially a mirror image on either side of a center point in sequence. So where there's a T here, there's an A here. Where there's a G here, there's a C here. Where there's a T here, there's an A here. As we branch out in either direction, we have a mirror image, mirror as in complementary sequences. These kinds of proteins can either block or promote the activity of RNA polymerase. Activator and repressor proteins bind to sequences on the DNA that are referred to as activator binding sequences or operators, respectively. So activators bind to activator binding sequences, and repressors bind to operator sequences, usually just referred to as operators. They act in association with small non-protein molecules called effectors. Okay, these are non-protein molecules. So an example of an effector is allolactose. That is relevant to the lac operon, which we'll discuss in a moment. Effectors can be co-repressors or inducers. And we'll look at some examples that should illustrate what those terms mean. Let's consider the concept of enzyme repression and induction, beginning with the figure on the right here. Beta-galactosidase is in the same family of enzymes referred to in humans as lactase. It is the enzyme, in E. coli at least, encoded by the LAC-Z structural gene found in the lac operon of many bacteria, most notably E. coli. You should be familiar with how the lac operon is regulated from earlier coursework, and if not, you should take a moment and review your notes from genetics class. In the lac operon, the transcription of the gene for beta-galactosidase is induced by the presence of allolactose. Allolactose is a breakdown product of lactose itself. So basically, if you have lactose present, that's what's shown here, then that means that you will have allolactose. And it's the allolactose itself that acts as the effector molecule. So allolactose is an effector molecule, and in this example, and in that operon, it is an effector molecule that serves a role as an inducer, right? It's an inducer type of effector molecule. Note that what happens if you track the blue line, which is depicting beta-galactosidase activity or level, that once you add lactose, which means that you've added allolactose, you see an increase in beta-galactosidase level. So the allolactose is acting as an inducer, it's increasing the level of beta-galactosidase. You'll note that the protein and cell number data, which are shown here and here, indicate that prior to the addition of lactose, there was some other carbon source present because protein and cell number were increasing over time. On the left, we see a scenario where the blue line represents, this is a different enzymatic system, the blue line represents the level of arginine, biosynthesis enzymes. Remember, arginine is an amino acid. So these are enzymes that are found in an anabolic pathway, the pathway required for making arginine. In this scenario, when arginine is added, the level of arginine biosynthesis enzymes plateaus. So the addition of arginine caused the level of that particular protein to plateau, to stop increasing. This indicates that arginine is acting as an effector molecule and that it is acting as a co-repressor type of effector molecule. Let's consider the regulation of these genes, those for LAC-Z, those that encode the beta-galactosidase, the LAC-Z gene, and also those that are important for arginine biosynthesis. Here is a diagram of the arginine biosynthesis operon. Remember that an operon is a group of genes located side by side, all regulated by the same promoter, and all transcribed consecutively, producing one piece of messenger RNA that is the primary transcript. This is then cut into individual messenger RNA transcripts for translation. So shown here is the arginine biosynthesis operator. These genes, ARG-C, ARG-B, and ARG-H, are all controlled by this ARG promoter. Okay, so when that promoter, when RNA polymerase, shown here, binds to that promoter, it can then transcribe all three of these genes consecutively, resulting in one primary messenger RNA transcript that would then be cut apart into the messenger RNA for ARG-C, ARG-B, and ARG-H respectively, and then they would go off to be translated. So the genes C, B, and H here are the structural genes that encode the enzymes necessary for arginine biosynthesis. The promoter is shown in yellow. RNA polymerase binds to that promoter, but it can only transcribe if the repressor protein, shown here and here, is not attached to the ARG operator sequence. The repressor protein's default state is to be off or unattached to the operator, as in the top of this figure, shown up here. Okay? So when the repressor protein is off the operator, transcription can proceed because there's nothing blocking RNA polymerase. However, if arginine is present, and arginine is shown here as this small pink ball, then the arginine will bind to the repressor protein, forming a complex that changes the conformation of the repressor protein, causing it to then be attracted to and bind to the ARG operator, as shown down here in the bottom part of the figure. This then effectively blocks transcription. So arginine is acting as an effector molecule, and it's the type of effector molecule called a co-repressor. That's a kind of molecule that when bound to a repressor acts to help repression occur, acts to block transcription. This way, when the cell has enough arginine, it won't continue to make more. So the obvious benefit energetically makes understanding these sorts of regulatory systems very easy and very intuitive. All right, now here is your old friend, the lac operon, which you should have learned all about in genetics. So the regulation of the lac operon is more complex than what is shown here. This is just one aspect of it, but we'll get to the other part in the next section of this lecture. The three structural genes of the lac operon are the LAC-Z, which encodes beta-galactosidase, the LAC-Y, which encodes a permease specific to the transport of lactose, so it's what gets lactose into the cells, and the LAC-A encodes an acetylase enzyme. The role of that enzyme in lactose degradation is not well understood, but we do know that lactose degradation won't happen in its absence. And the LAC-Z, as I said before, is the beta-galactosidase. Those are the structural genes of the lac operon. So lac operon is regulated similarly to the ARG operon we just looked at, but a little bit differently. In the case of the lac operon, there's another gene not shown here that encodes the repressor protein. and that other gene is constitutively expressed, so it is constantly producing this repressor protein. The default case in this operon is for the repressor protein to remain associated with or attached to the lac operator, okay, unless the inducer molecule is present, and the inducer molecule is allolactose. Okay, it's a product of lactose. It's basically always found if you have lactose around. It's just part of the chemical, a little bit of chemical breakdown, not mediated biologically, so that if you've got lactose, you always have allolactose. If allolactose is present, it will bind to the repressor protein, and that will cause the repressor protein to dissociate or let go of the lac operator, as shown in this lower part of the figure. That thereby allows RNA polymerase to travel from the promoter down the lac operon, transcribing these structural genes. And once they are transcribed, you would have messenger RNA for each of them. That would be translated, and then you would have the ability to degrade lactose. So lactose basically turns on the ability for the cell to break it down and use it as an energy source. And that makes a lot of sense bioenergetically, because why would you bother making enzymes to break down a sugar if the sugar wasn't in the environment at all? So the previous two examples were examples wherein we had pre-transcriptional regulation of a microbial operon that relied on negative control utilizing repressor proteins. Here's an example of the regulation of a bacterial operon using what is called positive control and activator proteins. So this is the maltose catabolic operon. MAL-E, MAL-F, and MAL-G shown here are structural genes needed for E. coli and other bacteria to degrade the sugar maltose, using it as a carbon and energy source. Here's the MAL promoter that controls the expression of all of those genes. So here we have a scenario where we have an activator protein. So the yellow is the maltose activator protein, okay? And an activator protein typically works to associate with RNA polymerase, helping to tether it to the promoter region and facilitating transcription. But in this case, the maltose activator protein can only do its job if an inducer molecule is bound to it. So for most organisms, the inducer molecule is maltose. It's a story similar to the lac operon in that the sugar itself, the presence of the sugar, can actually turn on the transcription of the genes that are required for the catabolism of that particular sugar. So if the E. coli or other bacteria are in an environment where maltose is present, and it might want to eat that maltose, then the maltose itself will serve as an inducer molecule. It'll bind to this activator protein. Together they will tether RNA polymerase onto the promoter region, and once that happens, transcription will proceed. You'll get transcription of these structural genes, and you'll get maltose catabolism. Very elegant and, again, very simple and intuitive system. This is positive control because we have an activator protein. That ends the first part of this three-part lecture. Now please go on to part 2.